Awesome. Check, check, 1, 2. How's everyone doing? Great. Awesome. Well, thank you for that wonderful introduction, Rachel. So, considering we're in the Bromeliaceae Symposium, I won't give too much backstory into the great deal of diversity that Bromeliaceae has across taxonomic, morphologic, and ecological scales throughout the 80 genera and around 3,600 known species. And of course, with all this diversity, there's many unanswered questions regarding the developmental, functional, and ecological trade-offs that impact reproductive features. And I hope that talks before and after mine throughout today's symposium, as well as tomorrow, help show you why this family is an ideal lineage to investigate some of these questions, evolutionary questions in reproductive biology, specifically regarding mating systems and pollination ecology. But it's all right. But if you don't trust me, then take a look at Google Scholar, and you'll find prominent figures in the Bromeliaceae community, also mentioning that this is a great group to study evolution. We have reviews, we have experimental studies, work being done, over 2 decades ago, and of course, more work being done in the past. But I wanted to highlight this recent review that came out by Cascante Marin and Nunez Hidalgo that was a very much needed new review, updated review of the review of breeding systems in this family. And one of the main takeaways is that there's not much knowledge in multiple groups and regions. It's very biased in the areas that we have a lot of data for breeding systems. And so I thought this would be a perfect segue into the data that I'll be talking about, which are from my undergraduate thesis, which had a main objective of understanding if outcrossing impacts reproductive investment in species that differed in reproductive biology. And so I did this in the context of life history, first by describing the natural history of these 2 plants, and then doing some crossing experiments. So the 2 species that I used were mentioned in Dr. Jabaily's talk earlier. So we have Vriesea rafaelii, which is in the subfamily Tillandsioideae, and then Billbergia brasiliensis in the subfamily Bromeliaceae. And they're both great species to do this work in because they're able to set seed in the greenhouse, but they differ in their reproductive traits. So Vriesea rafaelii flowers have an inconspicuous calyx that's the same colors as the bract and the scape, that green color. They have a corolla that's primarily this yellowish white, and it has these purple apical margins. Spirals, it recoils into a spiral at anthesis. The calyx and scape are covered in trichomes, which give the appearance of this white lepidote surface, also on the scape as well. And the flowers are odorless with a small amount of nectar at the base of the ovary, and the inflorescence is pendant, so falling over. And so for this study, I included 17 individuals of Vriesea rafaelii and 14 individuals of Billbergia brasiliensis from that larger greenhouse population. And then these were split into an outcrossed and self group, which then I'd be either selfed or outcrossed. And so to evaluate the ability of both of these plants to self-pollinate themselves, or if they're self-compatible, we looked at the self-compatibility index, which is a pretty simple mathematical equation. So it's the proportion of the reproductive success of the seed set of hand-celled flowers over the reproductive success of hand-crossed flowers, and then this ranges from either 0, so that would be more a species that's self-incompatible, so it would be the case of a plant that doesn't set seeds in its self flowers, but then does in its cross flowers, so that number would be much higher on the bottom than on the top. And then if a species is self-compatible, the index would be closer to 1 in the case of the numbers of the reproductive success being similar between selfed and outcrossed flowers. And then to answer the main objective if outcrossing or selfing influenced the reproductive investment in both of these species, we ran a Mann-Whitney U test. And so moving into the results, let me first kind of lay out the template of how I'm going to be describing the results. So on our y-axis, we'll have either fruit mass or seed mass in grams. And then on our x-axis, we have the experimental trials. So for the Vriesea rafaelii, I denoted it with a bat because I also forgot to mention the flowers open at night for Vriesea rafaelii. And then for the self-pollinated flowers, I will denote that with a paintbrush. And then, so what we see for Vriesea rafaelii investment into fruit biomass, we don't see any difference between outcrossed and self-flowers, but interestingly so, we do see this, this bimodal pattern of fruit biomass in both self and outcrossed fruits. And this was actually just recently discovered when we plotted this with a race car plot on the right and then the scatter plots. So we're still gonna look and kind of investigate this if there's biological or experimental designs. What's the reasoning behind this bimodal pattern? And then when looking at Billbergia brasiliensis investment into fruit biomass, we see that the outcrossed flowers had marginally higher fruit mass than flowers that were selfed. And then switching pages to the investment into seed biomass, we see, so we're back to Vriesea rafaelii, and we have, once again, the outcrossed and then the selfed. We see that Vriesea seed mass did not differ significantly between fruits that were selfed or outcrossed. This is probably due to the tail of the zero tail of fruits that didn't set seed in both groups. But if you look at the medians, we see that there is a higher investment in the outcrossed fruits for Vriesea rafaelii. When moving over to Billbergia brasiliensis, we see that outcrossed fruits had higher seed mass than self flowers, which is very cool to see. And when looking at the self-compatibility index, after crunching the numbers, we see that both species are self-compatible. Once again, that's the equation of the self-compatibility index, so proportion of self flowers, reproductive success over cross flowers. And it was, good to see that both of these species are self-compatible, as we thought previously. But the lower selfing investment in both of these species, and especially in the case of seed mass for Billbergia brasiliensis, suggests that inbreeding depression is occurring and that pollinators are important for a high reproductive output for both plants. And so, kind of switching pages, I spent a lot of time with these plants. They found me at a very dark time, and I think a lot of our lives during the pandemic, so 2020 to 2021, when I wasn't seeing many folks. I was, either at home with the few people I lived with, or thankfully was in the greenhouse at day in, day out, as well as night in, night out for the case of Vriesea rafaelii, as you see over in this picture, even wearing my mask in the greenhouse when I was the only one in there. And so a lot of biologists throughout this conference have actually mentioned this kind of know to discover, or you need to really get to know your study system, and by looking and noting, seeing plants out in the wild or continuously seeing them in the greenhouse, you start to note variation, and it's really important to know your study system to then be able to understand some of the variation and understand the results that you're getting from your data. And so I wanted to quickly highlight some variation that I noted in both species. So in the case of Vriesea rafaelii, there is some variation in reproductive organ length. And so this really manifested itself in the case of hercogamy, which was the most common variation in Vriesea rafaelii flowers. And we saw this in 29 flowers across multiple individuals. And I wanted to define hercogamy, which was the spatial separation of the anthers and the stigma, which we can see in this picture right here. And that's, the hercogamy looks different, and seeing this in 29 flowers compared to most of the flowers with the stigma really just laying on top of those anthers there. When looking at the variation in Billbergia brasiliensis, there was some differences in scape length. So instead of the scapes fully elongating and becoming pendant, there were a couple of individuals where the scape just stopped pretty much growing and it didn't fully elongate. So it was tough for me as the resident hummingbird to observe these plants and to do crosses, and I'm sure that in the wild this would be hard for both pollinators and fruit dispersers to reach those fruits. And so when I was seeing all this variation in both species, it really got me thinking about what does the Selfian syndrome look like in Bromeliaceae broadly. And so obviously we all know that imitation is the biggest form of flattery, and so I've been recently decided to do like an upset graph, which if you were at Dr. Speck's talk yesterday, you might have seen a similar figure, which is a way to view interactions between more than 3 groups. So instead of using a Venn diagram, you can use something like upset graph, which has its own graphical interface you don't even need to program if you want to use this. They do have R, Java, Python packages as well. And so to quickly run you through what this graph is showing you, so these are the traits that I decided to look at, which are from the recent review. And so these 2 traits, reproductive separation, which I clumped together both hercogamy as well as dichogamy into this one single trait in this case is reproductive separation. And first, let me define what dichogamy is, which is the temporal separation of male and female organs maturing at different times. And what you would expect is that a species that is self-compatible would not have this reproductive separation. This dichogamy as well as hercogamy makes it hard for a plant to be able to self-fertilize itself. But let me take a step back and finish explaining this graph. So these dots indicate that interaction, that trait is present in that certain group of in this case, bromeliad species, which came from the review from the supplemental materials. And I only included species that did have data on the reproductive separation. So either there was data on hercogamy or dichogamy. And so we have the traits here, the interactions here. So if there is not a dot present, then that means that trait's not there. And so you can really interpret that as the opposite of whatever trait you see right here. So in the case of reproductive separation, if there is no dot there, then that means that separation either temporally or spatially, time or distance, is absent. In the case of a species being epiphytic, if the dot's not there, then that means it's primarily terrestrial. We have self-compatibility, which I used a cutoff of 0.3 in the self-compatibility index, which is used in other previous work in bromeliad breeding system work to indicate if it's above 0.3, then the species is self-compatible. Below 0.3, then the species is self-incompatible. And then looking at various pictures on GBIF, iNaturalist, and as well as the manuscripts that were used in the review, I looked to see if the species had a red floral display, which I was using as a proxy for like a punitive hummingbird pollinator, like a hummingbird pollination syndrome. Yeah. And so I wanted to, as you can see here with the purple bars showing you where we see interactions with species being self-compatible, we see that there's not really an end-all be-all syndrome of selfing. It's kind of spread out, even just with these 4 traits here. There's various ways that a species has in terms of the reproductive traits, what they look like. And so I wanted to just quickly highlight a couple of these for folks who maybe don't spend as much time looking at bromeliads as I do. And so what we have here is we have like the group that's got the most species. So we have 6 species that have reproductive separation. So in the case of the species of Catopsis, we have the species as protandrous. So the anthers develop or mature first. The species is epiphytic, and it is self-compatible, but it doesn't have that red foil display. We have the white corolla there. Then on the other side of the coin, we have a species like the species of Acmea that does not have reproductive separation, but has that red floral display and still epiphytic. And then we have the do-it-all, the species of Tillandsia that's is hercogamous and protandrous, as well as epiphytic and has that red floral display. And then lastly, I wanted to highlight the species of Whitmackia that is protogynous, so the gynoecium develops first, and then the species is self-compatible but is not epiphytic and doesn't have that red floral display. And so really the main takeaway that I wanted you all to get from this graph is, as I said earlier, that there seems to be many ways that plants are having this selfing syndrome, as well as it seems like with the case of reproductive separation, that mixed mating seems to be playing a big role. Like species are self-compatible, but they're opting to have traits that really promote outcrossing rather than promoting self-fertilization. And so this really, once again, got me thinking kind of on another hand, what role does the self-compatibility have in the diversification of this family? And so a lot of folks who just work on breeding systems broadly, Boris Ejik back in 2008 coined this idea that self-compatibility is a quote unquote evolutionary dead end, which what they mean by this is that lineages that are self-compatible have lower net diversification rates than lineages that are self-incompatible. But in the case of our very much loved Bromeliaceae, I wonder if that's really the case, especially in diverse clades such as the bromelioid tank epiphytes and the core tillandsioids. And I was actually just reading this recent manuscript that got published by Igor and his collaborators, and I was just looking at one of the plates, and it was just blew my mind to see this difference in hercogamy and these closely related species. And so I wonder how much reproductive assurance does self-compatibility provide in a group that also has other adaptive traits, such as clonality or this amazing graph from Givnish et al. That was published a little bit ago that shows some of the adaptive traits that have led to the radiation of Bromeliaceae. So in the case of this graph or figure here, if the arrows have 2 arrowheads, that means there's this bi-causality. So in the case of dry habitats, that leads to species having CAM. There's some correlation there and vice versa. So CAM species also kind of you find in dry habitats. And so I've been speculating for a little bit now, and I'm really going to speculate about what role does self-compatibility have? So if we compare to some of these other previously studied traits. So say we throw self-compatibility up there, maybe does being in a dry habitat kind of correlate with a species being self-compatible, and maybe does that lead to a species being more species rich? These are the questions that I'm very curious and hope to explore very soon. And mind you, we do know that self-compatibility is a syndrome of traits. So it's not just being self-compatible. It's maybe the reduction of hercogamy, for example, on this slide. And then that leads to a species being self-compatible. And maybe there's this correlation with self-compatibility and species richness. So these are the questions that are swirling around in my head when I'm taking a shower, when I'm sleeping, obviously in the lab as well. And I hope to start exploring these. But in order of just with the time that I have, I want to say thanks to many folks. So thanks to my undergraduate collaborators, Dr. Jabaily, Dr. Heschel, Dr. Bodine, as well as my funding sources, the Heavey Fund and the Keller Family Fund, as well as the Bromeliaceae Society International for the assistance to come out to this conference. And then a huge thanks to the Spec Lab for my new home where I'm at, where I'll start asking some of these questions, and of course to my family who have supported me for my whole time. So.
Thank you. We have time for a couple of questions. Can I go around?
Okay, congratulations. A beautiful talk, and I really like the mating system things. Have you ever think to include in your thoughts in your shower while you are showering that these mating systems also be important when we have many species together in the same environment and they need to protect themselves from heterospecific pollen?
Mm-hmm. Yeah, that's actually, I unfortunately did not mention that, but that's another important question. Like what does, sympatric species and self-compatibility, how might that play into species not hybridizing? And there's just many questions that, that also needs to be put together into this puzzle of diversity in certain clades.
Good. Just another question. I don't know if I passed and didn't understand. The Chilanteranthus you show was doing selfing.
Mm-hmm.
Was it?
Yes. Yeah. That was a self-compatible. Yeah.
Okay. What did you get this result from yourself?
No. This was from the review. I don't remember exactly what manuscript this was from, but
okay, because we published something and then it was not self-compatible.
Oh.
Yeah,
well,
maybe
then
we can talk about that later.
Yeah.
Okay, congratulations. I'll just be sure that I was understanding correctly.
Mm-hmm.
Any other? Mm-hmm.
Okay, thank you, Josh, for the nice talk, and congratulations for your enthusiasm. And you're not alone in the shower. Even 30 years taking shower, thinking about bromeliaceae.
Yeah.
And you see, you have a whole world ahead, and the experience we have in the observation of the flowers in not only in the greenhouses but especially in the field. We have very kind of problems like you showed us with the different development of the flowers. Mm-hmm. And sometimes we go to the field to observe the flowers, and we have many of these problems, and we have 2 or 3 only individuals to deal with. Mm-hmm. You have a full greenhouse to this, and we have many problems to have many individuals with different kind of flowering. We have Vriesea, for example. Sometimes we have 3 or 4 individuals in the field, and each one opens 1 or none flowers a day.
Yeah.
We have to deal with this kind of difficulty. And with Billbergia, if you arrive in the field, and the day after, we have no flowers at all.
Yeah.
And I think it's a very good strategy to start with the greenhouse plants, but in the same way as Clarice told you, when we have 1 bromeliad, we have lots of them. And Vriesea, we have, and always we have, especially in the Atlantic forest, we have sometimes 2, 3, 4 species with the same syndrome. Mm-hmm. Sometimes many related species, hummingbird pollinated, flowering at the same time. And the hybrids are the reality for the Vriesea. It is very interesting to always think about this. We're starting a project now with the pollination syndrome in a Vriesea community with different pollination syndromes to check the gene pollen flux among them. As I told you, we have you and our students that are trying to study this now, an entire world ahead, because Vriesea, especially Vriesea, they are very different. At the same time, we have lots of species very similar. We have so many species, and the question is, the main question is, what's the species in Vriesea? That's our main question. Yeah. Okay, and think about the hybrids always, and go to the fields as soon as you can. We are ready to have you there with us.
Thank you for your insights, Andrea.
Okay, thank you very much.
Yeah,
congratulations for the work.
Cheers. I guess we'll chat later, but I have many things to say about that. So,
thank you very much.
Mm-hmm.